I'm Harry Glurkian, and this is Moneyball Medicine, the interview podcast where we meet researchers, entrepreneurs, and physicians who are using the power of data to improve patient health and make healthcare delivery more efficient. You can think of each episode as a new chapter in the never-ending audio version of my 2017 book, Moneyball Medicine, Thriving in the New Data-Driven Healthcare Market. Kevin, welcome to the show.
Great to see you again, Harry. Thanks for having me on.
Yeah, no, I mean, it's well, I seem to be saying this a lot lately. It's like, it's been such a long time since, because of this whole pandemic, nobody's really seeing anybody on a regular basis.
Yeah, that's right.
You know, I want to give everybody like a chance to hear about your, you had written this book called Editing Humanity, which is, beautifully placed behind you for product placement here. But I want to hear, like, can you give everybody sort of an overview of the book and why you feel that this, fairly technical laboratory tool called CRISPR is so important that you need to write a book about it?
Yep. Thank you. Yes. As you may know from some of my previous bestsellers or not, I've written about big stories in genetics because that's the only thing I'm remotely qualified to write about. I trained as a human geneticist in London and came over to do actually a pair of postdocs in the Boston area before realizing my talents, whatever they might be, certainly weren't as a bench researcher. So I had to find another way to stay in science, but get away from the bench and hang up the lab coat. So moving into science publishing and getting a job with Nature and then launching Nature Genetics was the route for me. And over the last 30 years, I've written 4 or 5 books that have all been about A, something big happening in genomics, B, something really big that will have both medical and societal significance, the mapping and discovery of the BRCA1 breast cancer gene in the mid-90s, the Human Genome Project at the turn of the century, and then the birth and the dawn of consumer genetics and personalized medicine with the $1,000 genome. And the third ingredient I really look for, if I'm trying to reach a moderately or significantly large audience, is for the human element, who are the heroes and the anti-heroes to propel the story. Where is the human drama? Because we all love a good, juicy, gossipy piece of story and rating the good guys and the bad guys. And CRISPR, when it first really took off in 2012, 2013, as a gene editing tool, a lot of scientists knew about this. I mean, these papers were being published in Science in particular, not exactly a specialized journal, but I was off doing other things and really missed the initial excitement, I'm embarrassed to say. It was only a couple of years later, working on a sequel to Jim Watson's DNA, where I was tasked with trying to find and summarize the big advances in genomic technology over the previous decade or whatever, that I thought, well, this CRISPR thing seems to be taking off, and the Doudnas and Charpentiers are winning breakthrough prizes and being feted by celebrities, and it's going on 60 Minutes. They're going to make a film with The Rock, Dwayne Johnson. What the heck is going on? And it took very little time after that for me to think, this is such an exciting, game-changing, disruptive technology that I've got to do 2 things. I've got to A, write a book, and B, launch a journal. And that's what I did and started planning at any rate in sort of 2016 and 17. We launched the CRISPR Journal at the beginning of 2018, and the book Editing Humanity came out towards the end of 2020, literally one day before the Nobel Prizes. How about that for timing?
Right.
For Doudna and Charpentier for chemistry last year.
Because when I think about it, like I remember, working with different companies that had different types of gene editing technology working with some, particularly in the sort of agriculture space, because it's
Yeah.
a little bit easier to run faster than in the human space.
Yeah.
And you could see what was happening. But CRISPR now is still very new, but from the news and different advances that are happening, especially here in the Boston area, it's having some real world impacts. I mean, if you had to point to the best or the most exciting example of CRISPR technology helping an actual patient,
Yep.
would you say, and I've heard you say Victoria Gray, I think,
Yep.
would be the person that comes to mind.
Yep.
I think in one of your last interviews, you said something about her being, her name will go down in history. Can you explain the technology that is helping her and what some of the similar uses of CRISPR might be?
Yeah. So I spent the first half of Editing Humanities is about the heroes of CRISPR, how scientists turned it from this bizarre, underappreciated bacterial antiviral defense system and leveraged it and got to grips with it, and then figured out ways to turn it into a programmable gene editing technology. And within a year or two of that happening, the classic Doudna Charpentier paper came out in the summer of 2012. Of course, the first wave of biotech companies were launched by some of the big names, indeed most of the big names in the CRISPR gene editing hierarchy. So Emmanuel Charpentier, Nobel laureate, launched CRISPR Therapeutics. Jennifer Doudna co-founded Editas Medicine with several other luminaries. That didn't go well for reasons of intellectual property, so she withdrew from Editas and became a co-founder of Intellia Therapeutics, as well as her own company Caribou, which just went public. And Feng Zhang and others launched Editas Medicine. So we had this sort of three-way race, if you will, by three CRISPR empowered gene editing companies who all went public within the next 2 or 3 years, and all set their sights on various different genetic Mendelian disorders with a view to trying to produce clinical success for this very powerful gene editing tool. And so, yes, Victoria Gray is the first patient, the first American patient with sickle cell anemia in a trial that is being run by CRISPR Therapeutics in close association with Vertex Pharmaceuticals. And that breakthrough paper, as I think many of your listeners will know, came out right at the end of 2020, published in the New England Journal of Medicine. Doesn't get much more prestigious than that. Right. And in the first handful of patients that CRISPR has edited with a view to CRISPR Therapeutics have edited with a view to raising the levels of fetal globin to compensate for the defective beta globin that these patients have inherited. The results were truly spectacular. And if we fast forward now to about 2 years after the initial administration, the initial procedures for Victoria Gray and some of her other volunteer patients, the results still look as spectacular. Earlier this year, CRISPR Therapeutics put out a sort of an update on where they are, saying that the first 20 or 24 patients that they have dosed with sickle cell and beta thalassemia are all doing well. There have been little or no adverse events. And the idea of this being a once and done therapy appear very well founded. Now, it's not a trivial therapy. This is ex vivo gene editing. There's obviously rounds of chemotherapy to provide the room for the gene edited stem cells to be re-implanted into the patient. So this is not an easily scalable or affordable or ideal system. But when were we ever able to say we've got pretty much a cure for sickle cell disease? This is an absolutely spectacular moment, not just for CRISPR, but for medicine, I think, overall. And Victoria Gray, who's been brilliantly profiled in a long-running series on National Public Radio, led by the science broadcaster Rob Stein. She is, we can call her Queen Victoria, we can call her many things, but I really hope, and that it's not just my idea, that she will be one of those names like Louise Brown and other heroes of modern medicine that we look and celebrate for decades to come. So the sickle cell results have been great. And then much more recently, also in the New England Journal, we have work led by Verve Therapeutics, one of the other 3 companies that I named, where they've been also using CRISPR gene editing, but they've been looking at a rare liver disease, a form of amyloidosis, where a toxic protein builds up, and looking to find ways to knock out the production of that abnormal gene. And to do so, they've been doing in vivo gene editing, really for using CRISPR for the first time. It's been attempted using other gene editing platforms like zinc fingers, but this is the first time that I think we can really say, and the New England Journal results prove it, in the first 6 patients that have been reported, remarkable reductions in the level of this toxic protein, not far better, but certainly better than any approved drugs that are currently on the market. So again, this is a very, very exciting proof of principle for in vivo gene editing, which is important not just for patients with this rare liver disorder, but it really gives, I think, the whole field and the whole industry enormous confidence that CRISPR is safe and can be used for a growing fraction of that, the list of Mendelian disorders. It's 6,000 or 7,000 diseases about which we know the root genetic cause. And we're not going to tackle all of them anytime soon, but the list of ones that now are within reach, and more companies are being launched all the time to try and get at some of these diseases. So as we stand here in the summer of 2021, it's a really exciting time. But of course, the future looks very bright, but there's so much more to be done.
No, we're just at the beginning. I mean, I remember when I first saw this, my first question was off-target effects. Yep. Right? How are we going to manage that? How are they going to get it to that place that they need to get it to have it to that cell at that time in the right way to get it to do what it needs to do? And, all these sort of technical questions. But at the same time, I remember I'm trying to explain this to my friends who I'm like, you don't understand, like this can change everything. Yeah. And now a high school student, I say this to people and they look at me strangely. Yeah. A high school student can order it and it shows up at your house. And
well, this is why I think, and this is why one reason why CRISPR has become such an exciting story and receives the Nobel Prize 8 years after the sort of the launch publication of the first demonstration of it as a gene editing tool is it is so relatively easy to get to work. It's truly become a democratized or democratizing technology. You don't need $1 million Illumina sequencer or anything close. And so labs literally all around the world can do basic CRISPR experiments. Now, not everyone is going to be able to launch a clinical trial, but the technology is so universally used, and that means that advances in our understanding of the mechanisms, new tools for the CRISPR toolbox, new pathways, new targets, new software, new programs. They're all coming from all corners of the globe to help not just medicine but many other applications of CRISPR as well.
Yeah, I always joke about like there are things going on in high school biology classes now that weren't they were not available, when I was in college, and even when we were in industry. And now, what used to take an entire room, you can do, on a corner of a lab bench. Yeah. So as far as the industry goes, we mentioned 3 companies, but today there's probably a dozen or more CRISPR-based or gene editing-based biotech companies. More undoubtedly about going to be launched before the end of this year. I'm sure we'll spend a bit of time talking about
CRISPR 2.0. It seems too soon to be even thinking about a new and improved version of CRISPR. But I think there's a lot of excitement around also two other Boston-based companies, Beam Therapeutics in Cambridge and Verve Therapeutics, both of which are launching or commercializing base editing. So base editing is a tool developed from the lab of David Liu of Harvard and Broad Institute fame. And the early signs, again, this technology is only 5 or 6 years old, but the early signs of this are incredibly promising. David's team, academic team, had a paper in Nature earlier this year, really reporting successful base editing treatment of sickle cell disease in an animal model, not by raising the fetal globin levels, which was sort of the more indirect method that is working very well in the clinic, but by going right at the point mutation that results in sickle cell disease. And using, given the chemical repertoire of base editing is able to make specific single base changes. It can't do the full repertoire of single base changes, so there are some limitations on researchers' flexibility. So they were unable to flip the sickle cell variant back to the quote unquote wild type variant. But the change they were able to make is one that they can live with, we can live with, because it's a known benign variant, a very rare variant that has been observed in rare people around the world. So that's completely fine. It's the next best thing. And so that looks very promising. Beam Therapeutics, which is the company that David founded or co-founded, is trying a related approach, also going right at the sickle cell mutation. So, and there are other companies, including one that Matthew Porteous has recently founded and has gone public, called Graphite Bio. So this is an exciting time for a disease, sickle cell disease, that has been woefully neglected. I think you would agree, both in terms of basic research funding and medical prioritization and medical education. Now we have many shots on goal, and it's not a matter of one's going to win and the others are going to fall by the wayside. Hopefully there will be, just like we have many COVID vaccines, we'll hopefully have many strategies for tackling sickle cell disease, but they are going to be expensive. And I think you know the economics of this far better than I do, but I think that is the worry, that by analogy with gene therapies that have been recently approved, it's really exciting that we can now see the first quote unquote cures in the clinic. That's amazingly exciting. But if the price tag is going to be $1 million or $2 million when these things are finally approved, if and when, that's going to be a rather deflating moment. But given the extraordinary research resources that the CRISPRs and Intellias and Beams and Graphites are pouring into this research, obviously they've got to get some return back on their investment so that they can plow it back into the company to develop the next wave of gene editing therapies. So it's a predicament.
One of these days, maybe I have to have a show based on the financial parts of it, because there's a number of different ways to look at it. But just for the benefit of the listeners, right, who may not be experts, how would you explain CRISPR is different from, say, traditional gene therapies? And is CRISPR going to replace, older methods of gene therapy, or will they both have their place?
No, I think they'll both have their place. CRISPR and these newer gene editing tools, base editing, and another one called prime editing, which has a company behind it now called Prime Medicine, are able to affect specific DNA changes in the human genome. So if you can target CRISPR, which is an enzyme that cuts DNA together with a little program, the GPS signal is provided in the form of a short RNA molecule that tells the enzyme where to go in the genome. And then you can either, a couple of strategies, you can either cut the DNA at the appropriate target site because you want to inactivate that gene, or you just want to scramble the sequence because you want to completely squash the expression of that gene, or particularly using the newer forms of gene editing like base editing, you can make a more nuanced, specific precision edit with one big potential advantage in the safety profile, which is you're not completely cutting the DNA, you're just making a nick and then coaxing the cell's natural repair systems to make the change that you're able to prime. So there are many diseases where this is the way you want to go, but that does not in any way invalidate the great progress that we're making in traditional gene therapy. So for example, earlier today, I was recording an interview for one of my own programs with Lawrence Reed, the CEO of Decibel Therapeutics, which is looking at therapies for hearing loss, both genetic and other types of hearing disorders. And I pushed him on this. Aren't you joining the gene editing kind of wave? And he was very circumspect and said, no, we're very pleased and very happy with the results that we're getting using old-fashioned gene replacement therapy. These are recessive loss of function disorders, and all we need to do is get the expression of some of the gene back. So you don't necessarily need the fancy gene editing tools if you can just use an AAV vector and put the healthy gene back into the key cells in a year. So they're complementary approaches, which is great.
So, in this podcast, I try to have a central theme when I'm talking to people, the relationships of big data, computation, advances in new drugs and other ways to keep people happy. So, question-wise, if there's no question in my mind that the whole genomics revolution that started in the 90s, and I was happy to be at Applied Biosystems when we were doing that, would have been impossible, in the absence of the advances in computing speed, storage in the last 3 decades. I think computing was the thing that held up the whole human genome, which gave us the book of life that CRISPR is now allowing us to really edit.
Right.
But I wonder if you could bring sort of up to date and talk about the way CRISPRs and say computation are intertwined. What happens when you combine precision of an editing tool like CRISPR with the power of machine learning and AI tools to find, meaning and patterns and in that huge genetic ball?
Yeah, I'm going to tread carefully here, but I think we are seeing some papers from some really brilliant labs that are using some of the tools that you mentioned, AI and machine learning, with a view to better understanding and characterizing some of the properties and selection criteria of some of these gene editing tools. So you mentioned earlier, Harry, the need to look out for safety and minimize the concern of off-target effects. So I think by using some of these some algorithms and AI tools, researchers have made enormous strides in being able to design the programmable parts of the gene editing constructs in such a way that you increase the chances that they're going to go to the site that you want them to go to, and just not get hung up latching onto a very similar sequence that's just randomly cropped up on some of the other the dark side of the genome, the other across the nucleus over there. You don't want that to happen, and I don't know that anybody would claim that they have a fail-safe way to guarantee that could never happen. But the clinical results that we've seen and all the preclinical results are showing in more and more diseases that we've got the tools and learned enough now to almost completely minimize these safety concerns. But I think everyone, while they're excited and they're moving as fast as they can, they're also doing this responsibly. I mean, they have to, because no field, gene therapy or gene editing, really wants to revisit what happened, as you, many of your listeners know. You know where I'm going with this, of course, the Jesse Gelsinger tragedy in 1999, when a teenage volunteer died in volunteering for a gene therapy trial at Penn with somebody with a rare liver disease. And of course, that setback set back the entire field of gene therapy for a decade.
Yes.
And it's really remarkable that you know, many of the sort of pioneers in the field refused to throw in the towel. They realized that they had to kind of go back to the drawing board, look at the vectors again, throw out, not completely, but a lot of work with adenoviruses has now gone by the wayside. AAV is the new virus that we hear about. It's got a much better safety profile. It's got a smaller cargo hold, so that's one drawback, but there are ways around that. And the explosion of gene therapy trials that we're seeing now, largely on the back of AAV and now increasingly with non-viral delivery systems as well, is very, very gratifying. And it's really delivery, I think, that is now the pain point. Digressing from your question a little bit, but delivery, I think, is now the big challenge. It's one thing to contemplate a gene therapy for the eye, for a rare hereditary form of blindness, or the ear. Indeed, those are very attractive sites and targets for some of these early trials, because the quantities that you need to produce and the localization, the physical localization, those are good things. Those help you hit the target that you want to. But if you're contemplating trying something for Duchenne muscular dystrophy or spinal muscular atrophy or some of the diseases of the brain, then you're going to need you know, much higher quantities, particularly for muscular disorders, where you know, you run into now other challenges, including, you know, production, manufacturing challenges, and potentially safeguarding and making sure that there isn't an immune response as well. That's another issue that is always percolating in the background. But given where we were a few years ago and the clinical progress that we've talked about earlier on in the show, I think you can safely assume that we've collectively made enormous progress in negating most, if not all, of these potential safety issues.
No, and you know, it's funny. I know that people will say, like, you know, there was, you know, a problem in this and that, and I look at, like, we're going into uncharted territories, and there has to be expected that it's not even like you've got people that knew what they were doing. All these people are new at what they are doing. And so you have to expect that along the way, everything's not going to go perfectly. But I don't look at it as a negative. I look at it as a, you know, they're the new graduating class that's going to go on and understand what they did right or wrong, and then be able to modify it and make an improvement. And, you know, that's what we do in science.
Well, and forget gene editing, in any area of drug development and pharmaceutical delivery, things don't always go according to plan. I'm sure you've had many guests on Moneyball Medicine who have had to deal with clinical trial failures and withdrawing drugs that, you know, they had all kinds of high hopes for, because we didn't understand the biology, or there was some other reaction. We didn't understand the dosing. You can't just extrapolate from an animal model to humans. And on and on and on. And so gene editing, I don't think necessarily should be held to any higher standard. I think the CRISPR field has already, in terms of the sort of market performance of some of the companies that we've mentioned, oh my God, it's been a real roller coaster, not surprisingly, because every time there's been a paper published in a prominent journal that says, oh my God, there's a deletion pattern that we're seeing that we didn't anticipate, or we're seeing some immune responses, or we're seeing unusual off-target effects. All of these, we're seeing P53 activation. And, you know, those are at least 4 off the top of my head. I'm sure there have been others. And all had big transient impacts on the financial health of some of these companies. But I think that was to be expected. and the companies knew that this was just an overreaction. They've worked and demonstrated through peer review publications and preclinical and other reports that these challenges have been identified, were known about, and pretty much completely have been overcome or are in the process of being overcome. So, and we're still seeing in just traditional gene therapy technologies that have been around for 15, 20 years, we're still seeing, reports of adverse events in some of those trials. So for gene editing to have come as far as it's come, and to be able to look at these two big New England Journal success stories in sickle cell and ATTR amyloidosis, I don't think very few, except the most ardent evangelists, would have predicted we'd be where we are just a few years ago.
Yeah, no, well, the crazy of us that try to invest in this space, like you have to pray and hope that your hunch is right. But speaking of this, like one of your previous books was called The $1,000 Genome. And when you published it back in 2010, right? I'm sure if you even said to someone $1,000 genome, like they would have said, yeah, I'm not so sure, right? But companies like Illumina have now blown past that.
Yep.
They're, very quickly. People are talking about sequencing a few $100 genome. Yep. I think the CEO of Illumina recently, or last JP Morgan, I think, said we're shooting for a $60 genome. How much did computing contribute to that trend, do you think? And do you wish you had called the book maybe the $100 genome?
I've thought about putting out a sequel to the book. We're just scratching off one of the O's and hoping nobody would notice. Computing was, yes, of course, a massive. It was, for the very first human genome. Remember the struggles
yes.
to put that first assembly together. Yes, it's not just about the wet lab and pulling the DNA sequences off the machines, but then the rapid growth of the data explosion, the ability to store and share and send across to collaborators and put the assemblies together has been critical, absolutely critical to the development of genomics. I remember, speaking of people expressing shock at the $1,000 genome, I called the book that because I heard Craig Venter use that phrase in public for the first time in 2002, and I'd only just recently published Cracking the Genome, and we were all still recoiling at the billions of dollars it took to put that first reference genome sequence together. And then here's Craig Venter chairing a scientific conference in Boston, saying what we need is the $1,000 genome. And I almost fell off my chair, like, you're on Fantasy Island. There's no way we're still doing automated Sanger sequencing. God bless Fred Sanger. But how on earth are you going to take that technology and go from billions of dollars to a couple of $1,000s? This is insanity. That session, we had a 2002 Boston. He had a little episode of America's Got Talent, and he invited half a dozen scientists to come up and show what they had. And George Church was one of them. I think Applied Biosystems may have given some sort of talk during that session. And then a young British guy from a company we'd never heard of called Solexa showed up and showed a couple of pretty PowerPoint slides with colored beads representing the budding DNA sequence on some sort of chip. I don't know that he showed any data. It was all very pretty and all very fanciful. And well, guess what? They had the last laugh. Illumina bought that company in 2006. And as you said, Harry, I think when they first professed to have cracked the $1,000 genome barrier, a few people felt they needed a pinch of salt to go along with that. But I think now, we're well past that. And there are definitely outfits, BGI, the Beijing Genomics Institute being one of them, that are touting new technologies that can get us down to a couple of hundred. And those were such fun times, because for a while there, Illumina had enormous competition from companies like 454 and Helicos and PacBio. And it was really, those were fun, heady times with lots and lots of competition. And in a way, Illumina's had it a little easy, I think, over the last few years. But
yes,
with PacBio and Oxford Nanopore gaining maturity, both in terms of the technology platforms and their business strategy and growth, I think Illumina is going to start to feel a little bit more competition in the long read sequence space. And one is always hearing whispers of new companies that may potentially disrupt next-gen sequencing. And that would be exciting, because then we'd have an excuse to write another book.
Well, Kevin, start writing, because I actually think we're there.
Oh, yeah?
I think there are a number of things there, and you're right. I think Illumina has not had to bring the price down as quickly because there hasn't been competition.
Yeah.
And when I think about the space, is if you could do a $60 genome, right? It starts to become a rounding error. Like, what other business models and opportunities now come alive? And those are the things that excite me. Right. But you have a unique, position as editor of the Journal of CRISPR and the former editor of a lot of prominent, publications, Nature Genetics, Bio IT World, Chemical Engineering, and Engineering News. Do you think that there's adequate coverage of the biological versus the computing side of it? Because I have this feeling that the computing side still gets a little overlooked and underappreciated.
I think you're right. I mean, I think at my own company, Genetic Engineering News, we still, I think we have such deep roots in the wet lab vision and version of biotechnology, that it takes a conscious effort to look and say, that's not where all the innovation is happening. Bio IT World, which you mentioned, is interesting because we launched that in 2002 as part of, it was launched by the publisher IDG, best known for Macworld and Computer World, and this whole family of high-tech publications. And we launched in 2002 as a very thick, glossy print magazine. And ironically, we just couldn't find the advertising to sustain that effort, at least in the way that we'd envisioned it. And in 2006 and 2007, your friend and mine, Philip School, the proprietor of Cambridge Health Tech Institute, kind of put us out of our misery and said, you know what, I'll take the franchise, because IDG just didn't know what to do with it anymore. But what he really wanted was the trade show that we'd taken over production of. And even though the magazine eventually, we fell on our sword and eventually put it out of its misery, but the trade show went from strength to strength, and it'll be back in Boston very soon, because he had the vision to realize there is a big need here, a sort of a supercomputing for life sciences. And it's not just about the raw high performance computing, but it's about the software tools, and data sharing and management. And it's great to go back to that show and see the Googles and Amazons and all the big household names are all looking at this, because genome technology, as we've discussed earlier, has been one of the big growth boom areas for their services and their products.
Right. I mean, well, if you look at companies like Tempest, right? When I talked to Joel Dudley over there on the show, it's they want to be the Amazon AWS piping for all things genomic analysis, right? So instead of creating it on your own and building it, just use their platform, basically. So it's definitely a growth area, and at some point, if you have certain disease states, I don't see how you don't get, genomic sequencing done.
Right. Right.
How a physician, even today in oncology, how anybody can truly prescribe with all the drugs that are being approved that have,
Yeah.
genomic biomarkers associated with them and not use that data. I'm always a little,
Yeah.
Yeah.
I mean, I'm on a much lower lo-fi scale, as I've been doing a lot of reading about sickle cell disease lately. It's clear that a lot of patients who are, of course, as your listeners know, are mostly African American, because the disease arose in Africa, and the carrier status gives carriers a huge health advantage in warding off malaria. So the gene just continues to stay high in frequency. Many African American patients would benefit from some generic drugs that are available in this country that provide some relief, but aren't aware of it, and maybe their physicians aren't completely aware of it either, which is very sad. And we've neglected the funding of this disease over many decades, whereas a disease like cystic fibrosis, which affects primarily white people of Northern European descent, that receives far more funding per capita, per head, than a disease like sickle cell does. But hopefully that will begin to change as we see the potential of some of these more advanced therapies. I think, as far as your previous comment, I think one of the big challenges now is how we tackle common diseases. I think we're making so much progress in treating rare Mendelian diseases, and we know thousands of them. But it's mental illness and asthma and diabetes, diseases that affect millions of people, which have a much more complicated genetic and in part environmental basis. And what can we learn, to your point about having a full genome sequence, what can we glean from that will help the medical establishment diagnose and treat much more common diseases, not quite as simple as just treating a rare Mendelian version of those diseases. So that's, I think, going to be an important frontier over the next decade or two.
Yeah, complicated, I think. And where the best, I think, you're going to see as we get more real world data that's, organized and managed well, along with genomic data, I think you'll be able to make more sense about it. But some of these diseases are quite complicated. It's not going to be find one gene and it's going to give you that answer. But I want to go back to, you can't really talk about CRISPR without talking about this specter of germline editing. And a big part of your book is about this firestorm of criticism and condemnation around, the 2018, when the Chinese researcher Hu Zhuanghui, I think I said it correctly.
Zhuanghui is how I would, but close.
Announced that he had created a pair of twin baby girls with edits to their genomes that were intended to make them immune to HIV, which sort of that already made me go, what? But the experiment was, it seems, unauthorized. It seems that, from what I remember, the edits were sloppy. And the case spurred a huge global discussion about the ethics of using CRISPR to make edits that would be inherited by future generations. Now, where are we in that debate now? I mean, I know the National Academy of Sciences published a list of criteria which says, don't do that.
It was a little more nuanced than that. It wasn't don't do that. It was, there's a very small window through which we could move through if a whole raft of criteria are met. So they refused to say hereditary genome editing should be banned, or there should be a moratorium, but they said it should not proceed until we do many things. One was to make sure it is safe, and I think we can't we can't run before we can walk. And by that, I mean, we've got to first demonstrate, because shockingly, this hasn't been done yet, that genome editing can be done safely in human embryos. And in the last 18 months, there have been at least 3 groups, arguably the 3 leading groups in terms of looking at genetic changes in early human embryos: Kathy Nyken in London, Shukrat Mitalipov in Oregon, and Dieter Egli in New York, who all at roughly the same time published reports that said, or posted preprints at least, that said, when we attempt to do CRISPR editing experiments in very early human embryos, we're seeing a mess. We're seeing a slew of off-target and even on-target undesirable edits. And I think that says to me, we don't completely understand the molecular biology of DNA repair at the early human embryo. It may be that there are other factors that are used in embryogenesis that are not used in well, you know, after we're born. That's speculation on my part. I may be wrong, but the point is, we still have a lot to do to understand, even if we wanted to, and even if everybody said, here's a good case where we should pursue germline editing, we've got to be convinced that we can do it safely. And at the moment, I don't think anybody can say that. So that's a huge red flag. But let's assume, because I believe in the power of research, and let's assume that we're going to figure out ways to do this safely. Or maybe we say, you know, CRISPR isn't the right tool for human embryos, but other tools such that we've touched on earlier in the show, base editing or prime editing, or maybe CRISPR 3.0, whatever that is, that's about to be published somewhere. Those are more safer, more precise tools. Then we've got to figure out, well, under what circumstances would we even want to go down this road? And the pushback was quite rightly that, well, we already have technologies that can safeguard against families having children with genetic diseases. It's called IVF and pre-implantation genetic diagnosis. So we can select from a pool of IVF embryos, the embryos that we can see by biopsy are safe and can therefore be transplanted back into the mother, taken to term, and you know, a healthy baby will emerge. So why talk about gene editing when we have that proven technology? And I think that's a very strong case. But there are a small number of circumstances in which pre-implantation genetic diagnosis will simply not work. And those are those rare instances where a couple who want to have a biological child, but both of them have a serious recessive genetic disease. Sickle cell would be an obvious case in point. So if 2 sickle cell patients who by definition carry 2 copies each of the sickle cell gene want to have a healthy biological child, pre-implantation genetic diagnosis is not going to help them because there are no healthy embryos from whatever pool that they produce that they can select. So gene editing would be their only hope in that circumstance. And the National Academy's report that you cited, Harry, did say for serious diseases such as sickle cell and maybe a few others, they could down the road potentially see and condone the use of germline gene editing in those rare cases, but they're going to be very rare, I think. So it's not possible that in an authorized, approved setting that we will see the return of genome editing. But that, of course, you can issue no end of blue ribbon reports from all the world's experts, and that's not going to necessarily prevent some entrepreneur whose ethical values don't align with yours or mine to say, you know what, there's big money to be made here. I'm going offshore and I'm going to launch a CRISPR clinic, and you know, who's going to stop me? Because I'll be out of the clutches of the authorities. And I think a lot of people are potentially worried that that scenario might happen. Although if anyone did try to do that, they would have, I mean, the scientific establishment would come down on them like a ton of bricks, and there'd be a lot of pressure brought to bear, I think, to make sure that they didn't cause any harm.
Yeah, it's funny. I would like to not call them entrepreneurs. I like entrepreneurs. I'd like to call them a rogue scientist.
Yeah,
that would be.
Well, I spent a lot of time. I spent, so there, as you say, there's the third of section of 4 in editing humanity. was all about the He Jiankui debacle or saga, I had flown to Hong Kong. It's a funny story. I had a little bit of money left in my travel budget, and there were two conferences in one in Hong Kong and one in China coming up in the last quarter of 2018. So I thought, well, I'll go to one of them. And I just, for narrow, almost a flip of a coin, I thought, okay, let's go to the Hong Kong meeting. It's a bioethics conference. I don't expect it to be wildly exciting, but there are some big name speakers, and this is an important field for the CRISPR journal to monitor. So flew there, literally, try to get some sleep on the long flight from New York, and then on landing, turn on the phone, wait for the new Wi-Fi, the new wireless signal provider to kick in, and then saw Twitter just explode on my feed as this very, very astute journalist at MIT Technology Review, Antonio Regalado, had really got the scoop of the century by identifying a registration on a Chinese clinical trial website that he, and only he, had the foresight and intelligence to sort of see, based on, he'd met He Jiankui in an off-the-record meeting, as I describe in the book, about a month earlier. His spider sense was tingling. He knew something was up. And this was the sort of the final clue. He didn't know at that time that the Lulu and Nana, the CRISPR babies that you mentioned, had actually been born, but he knew that there was a pregnancy, at least one pregnancy, from some of the records that he'd seen attached to this registration document. So it's a brilliant piece of sleuthing. And what he didn't know is that the Associated Press chief medical writer, Marilyn Marchioni, had confidentially been alerted to the potential upcoming birth of these twins by an American PR professional who was working with He Jiankui in Shenzhen. So she had been working on an embargoed big feature story that He Jiankui and his associate hoped would be the definitive story that would tell the world, we did this, quote unquote, responsibly and accurately, and this is the story that you can believe. So that story was posted within hours. And of course, the famous YouTube videos that He Jiankui had recorded, announcing with some paternal pride that he had ushered into the world these two gene-edited children, screaming and crying into the world as beautiful babies, I think was. And he thought that he was going to become famous and celebrated and lauded by not just the Chinese scientific community, but by the world community for having the ability and the bravery to go ahead and do this work after Chinese researchers had spent the previous few years editing human embryos. And when he was persuaded that he had to present his work in Hong Kong because he'd set off such an extraordinary firestorm. And I think you've all seen now the clips of the videos of him nervously walking onto stage, the muffled, the silence, the only sound, I was in the front row, the only sound in this big auditorium at Hong Kong University was absolutely packed to the rim. One side of the auditorium was packed with press photographers, hundreds of journalists, and cameras clicking and the shutters clattering that was the applause that he got as he walked on stage. And to his credit, tried to answer questions directly in the face of great skepticism from the audience. The first question of which was posed by David Liu, who had traveled all the way there, who just asked him simply, what was the unmet medical need that you were trying to solve with this reckless experiment? There are medical steps that you can do, even if the couple that you're trying to help has HIV and you're trying to prevent this from being passed on. There are techniques that you can use, sperm washing being one of them, that is a key element of the IVF process, to ensure that no HIV is transmitted. But he was unable to answer the question in terms of, I'm trying to help a family. He'd already moved out and was thinking far, far bigger.
Right.
And his naivete was shown in the manuscript that he'd written up and by that point submitted to Nature excerpts of which were leaked out sometime later. So he went back to Shenzhen. He was put under house arrest after he gave that talk in Hong Kong, and about a year later was sentenced to 3 years in jail. And so he's, to the best of my knowledge, that's where he is. But I often get asked, what about the children? We, as far as we know, there was a third child born about 6 months later, also gene edited. We don't even know a name for that child, let alone anything about their health. So one hopes that somebody in the Chinese medical establishment is looking after these kids and monitoring them and doing appropriate tests. The editing, as you said, was very shoddily performed. He knocked out the gene in question, but he did not mimic the natural 32 base deletion in this gene CCR5 that exists in many members of the population that confers essentially HIV resistance. So Lulu and Nana and the third child are walking human experiments. Sad to say, this should never have been done, never should have been attempted. And so we hope that he hasn't condemned them to a life of, cancer checkups, and that there were no off-target effects. They'll be able to live with this, hopefully, with this inactivated CCR5 gene, but it's been inactivated in a way that I don't think no other humans have ever been recorded with such modifications. So we really hope and pray that no other damage has been done.
So before we end, I'd love to give you the chance to speculate, say, on the future of medicine in light of CRISPR. Easy, fast, inexpensive genome sequencing, give us access to, let's say everybody's genetic code if they so chose. Machine learning, other forms of AI are helping understand the code and trace interaction between our 20,000 genes, and now CRISPR's gives us a way to modify it. So, it feels like we're almost everything we need to create, precise, targeted, custom cures for people with genetic conditions. What might be possible soon in your view? What remaining problems need to be solved to get to this new area of medicine?
If you know the sequence that has been mutated to give rise to a particular disease, then in principle, we can devise some sort of gene edit to repair that sequence. It may be flipping the actual base or bases directly, or maybe, as we saw with the first sickle cell trial, it's because we understand the bigger genetic pathway. We don't have to necessarily go after the gene mutation directly, but there may be other ways that we can compensate, boost the level of a compensating gene. But I think we should be careful not to get too carried away. As excited as I am, and hopefully my excitement comes through in editing humanity, but for every company that we've just mentioned, you can go on their website and look at their pipeline. And so there, Editas might have maybe 10 diseases in its crosshairs, and CRISPR might have 12 diseases, and Intellia might have 14 diseases, and Graphite has got maybe a couple, and Beam Therapeutics has got maybe 10 or 12, and Prime Medicine hasn't listed any yet, but will hopefully have a few announced soon. And so how many have I just reeled off? 50, 60, less than 100. And some of these are going to work really, really well, and some aren't going to be either proven ineffective or unviable economically, because the patient pool is too small. And we've got, how many did we say? 6,000 known genetic diseases. Right. So one of the companies that is particularly interesting, although they would admit they're in very early days yet, is Verve Therapeutics. I touched on them earlier because they're looking to modify a gene called PCSK9 that is relevant to heart disease, and could be a gene modification that many people might undergo because their PCSK9 gene may be perfectly fine and the sequence could be perfectly normal. But we know that if we remove this gene, levels of bad cholesterol plummet, and that's usually a good thing as far as heart management goes. So,
Yep.
so that's an interesting, very interesting case study, I think, to monitor over the coming years, because there's a company looking at a much larger patient pool potentially than just some of these rare syndromes with unpronounceable names. So the future for CRISPR and gene editing is very bright. I think one of the lessons I took away from CRISPR in editing humanity is looking at the full story, is how this technology, this game-changing gene editing technology developed, because 25 years ago, a handful of European microbiologists got really interested in why certain microbes were thriving in a salt lake in southeastern Spain. This is not exactly high profile, NIH must fund this research. There's a biological question that they wanted to answer. And the CRISPR repeats and the function of those repeats fell out of that pure curiosity, just science for science's sake. And so it's the value of basic investigator-driven, hypothesis-driven research that led to CRISPR being described, and then the function of the repeats, and then the story shifted to a yogurt company in Europe that was able to experimentally show how having the right sequence within the CRISPR array could safeguard their cultures against viral infection. And then 5 years of work, people in various groups started to see were drawn to this like moths to a flame. Jennifer Doudna was intrigued by this from a tip-off from a coffee morning discussion with one of her Berkeley faculty colleagues, Jill Banfield, a brilliant microbiologist in her own right. And then she meets Emmanuel Charpentier in Puerto Rico at a conference, and they strike up a friendship and a collaboration over the course of an afternoon. And why should that have worked? Well, it did, because a year later they're publishing in Science. So it's serendipity and basic research. And if that can work for CRISPR, then I know that there's another technology, beginning to emerge from somewhere that may, could yet trump CRISPR. And I think the beauty of CRISPR, with its universal appeal and the fact it's drawn so many people, is that it could be in Japan or China or South Korea or parts of Europe or Canada or the US or South America. Somebody is taking the elements of CRISPR and thinking, well, how can we improve it? How can we tweak it? And so this CRISPR toolbox is being expanded and modified and updated all the time. So it's a hugely exciting future for genome editing. And whether it's a new form of sequencing or a new form of synthetic biology, hopefully your show is going to be filled for many years to come with cool, talented, young, energetic entrepreneurs who've developed more cool gadgets to work with our genome and other genomes as well. We haven't even had time to talk about what this could do for plants.
Yeah, that's true.
Or rescuing the woolly mammoth from extinction. So all
yes.
fun things, but maybe another time.
Excellent. Well, great to have you on the show. Really appreciate the time. I hope everybody got a flavor for the enormous impact this technology can have on, like you said, we talked about human genome, but there's so many other genomic applications of CRISPR that we didn't even touch on.
Yep. So you have to read the book.
Yeah, I will look forward to the next book. So great. Thank you so much.
Thanks for having me on the show, Harry. All the best.
Take care.